Hello, Mrs. Anderson. My name is Dr. Braga. I'll be taking care of you today. Can you please verify your name and date of birth?
Ava Anderson, September 2nd, 1998.
Thank you. And what brings you in today?
A week ago, I returned home from a 2-week-long camping trip from New Hampshire, and I began to have symptoms like a headache, fatigue, fever, muscle and joint aches, and a rash on my inner right thigh. Surprisingly, I am not experiencing any pain or itchiness from my rash, however. It was concerning me.
Could I take a look at your rash?
Yep.
Okay, I see. I'm going to ask some questions so I can gather more information, understand your situation, and determine how we're going to treat it, okay?
Okay.
Can you describe the environment where you were camping?
It was a wooded area. My friends and I found this beautiful trail area. However, the trail wasn't clear, so we went to some grassy area to pass.
And do you remember finding ticks on your skin while hiking on the trail?
It must have attached itself while I was hiking. However, I only found it when I came back home and was changing my clothes.
Okay, and do you recall how it was removed from your thigh?
I used my bare hands and flicked it quickly because I was scared.
And did you rinse the area where it was attached?
No, but now looking back, I could have gone in the shower immediately to clean myself.
And do you remember how long this tick was on you?
No, I don't remember. Since we were hiking most of the whole day, I only noticed the day after. I'm not sure if it was attached in the beginning or after the hiking trip.
When did you first start noticing fatigue, headache, and fever and your other symptoms?
I began to feel these symptoms a day after I returned home. I started to feel muscle and joint aches and noticed 2 huge rashes on my right inner thigh.
Can you describe how the rash started and if the size changed?
It began to be 2 small red ovals but expanded significantly after a few days.
Did you wear protective clothes during your hiking trip?
I wore a t-shirt and shorts with long socks below my knees and shoes proper for hiking.
Have you had any illnesses recently or traveled anywhere outside of the States?
No.
Do you have any allergies, especially insect bites? And are you pregnant and on birth control?
No, I don't have any allergies including insect bites, and no, I'm not pregnant nor on birth control.
Okay, thank you. Based on your answers, the rash suggests you have erythema migrans since it has a bold eye shape, and your symptoms are often associated with Lyme disease. Lyme disease is caused by a bacterium called Borrelia burgdorferi, which can be transmitted through tick bites. Individuals have a higher chance of getting affected if the tick is attached for a long time. That's why I asked how long it was attached to you, and considering it was not properly removed, you possibly have Lyme disease. As you told me, you quickly removed it with your bare hands. To remove the tick properly, you should grasp the tick firmly and as close to the skin as possible with a steady motion. Then rinse it with rubbing alcohol to the skin as possible or soap and water. The following steps I will take to confirm the diagnosis and treat you effectively are gram staining, a lactose fermentation test, and an antibiotic resistance test. Do you have any questions for me?
If I do have Lyme disease, is it dangerous?
It can be dangerous if left untreated and has long-term symptoms, but I will conduct these tests to see if it is Lyme disease and provide you with an effective treatment. But Lyme disease is rarely fatal. I will take some samples from you and share the results through my chart. As of right now, keep an eye on any symptoms, and if you experience any worsening symptoms, please let me know immediately. Try to keep the area of the rash clean and dry without scratching it. If you don't have any more questions, you can return home and rest.
Thank you, Dr. Braga. I will take a look at it. I'll be waiting for a response then.
The first test I will conduct is a gram staining test. This test will help identify the bacteria causing Mrs. Anderson's infection and simultaneously identify the morphology and arrangement of the bacteria. Different types of bacteria have different cell structures and are divided into 2 groups, gram-negative and gram-positive. These groups are categorized based on differences in the bacterial cell wall structure. Gram-positive bacteria have a thick peptidoglycan layer, lack an outer membrane, and contain lipoteichoic acids. Gram-negative bacteria have a thin peptidoglycan layer consisting of an outer membrane and lipopolysaccharide A, known as LPS. It is crucial to know the type of bacteria because antibiotics function differently for each group, indicating that bacteria have different susceptibility to antibiotics. For further explanation, antibiotics that treat gram-positive bacteria will target the cell wall, whereas antibiotics that treat gram-negative bacteria will target the outer membrane first. I will take a sample and perform 2 gram stains in the clinical laboratory because gram-positive bacteria retain crystal violet dye, while gram-negative bacteria are counterstained with safranin, since it does not retain crystal violet dye. This will determine if the bacteria are gram-negative, which stains pink, or gram-positive, which stains purple. This will help me narrow down the most effective antibiotic for treatment. If the results become in gram-negative bacterium by picking up the counterstain, it would further support the identification of Borrelia burgdorferi, as it is a gram-negative bacterium. Fortunately, we have a well-equipped lab, so the results are usually available with analysis in a few hours, and I will send Mrs. Anderson's result through my chart. The second exam I'll conduct is a lactose fermentation test using a selective growth media called macaque agar plate. This plate is selected for gram-negative bacteria, and it is helpful to narrow down the bacteria by preventing the growth of gram-positive bacteria. Considering that the macaque agar plate contains 2 selective agents, bile salts and crystal violet, and inhibits the growth of gram-positive bacteria. This is important since Borrelia burgdorferi is a gram-negative bacterium. In terms of the process, I'll be using a macaque agar plate and a sterile wooden stick to inoculate the plate. If the bacteria cannot ferment lactose, the plate will turn brown, which is a feature of Borrelia burgdorferi. If the bacteria can ferment lactose, the plate will turn red or pink. Therefore, if it turns brown, it could be Borrelia burgdorferi. To prepare for this test, I'll perform a quadrant streaking technique to isolate and spread bacterial cells across the agar plate. To do this, I'll inoculate a small sample of the bacteria and begin streaking from one quadrant to the other to isolate colony growth. The agar plate will be incubated in their appropriate condition, which is our body temperature, 37 degrees Celsius. Considering this will be incubated for a few days, I'll contact Mrs. Anderson through my chart to send the results and provide her with effective treatment. The last exam I'll conduct is a Kirby-Bauer test, in other words, an antibiotic resistance test. This test will allow me to see the best treatment for the bacterial infection. I'll take a sample from Mrs. Anderson's rash to grow the bacteria, test its sensitivity to different antibiotics, and incubate it for 24 hours at 37 degrees Celsius. I'll measure the zone of inhibition in millimeters by looking at the diameter around each antibiotic disc. Afterward, I will compare the zone diameters to the Kirby-Bauer interpretive chart. If it is sensitive, indicates it will kill Borrelia burgdorferi, whereas if it is non-sensitive, it cannot kill the bacteria. From there, based on her clinical history, I will choose the top 3 most effective antibiotics appropriate for Mrs. Anderson. Considering she is not pregnant, not on birth controls, nor has allergies, it gives me a better chance to prescribe an effective antibiotic with no contraindications. This process takes overnight. So once the results are ready, I'll contact Mrs. Anderson through her MyChart with the results and prescribe the most appropriate antibiotic. I will guarantee the prescription is sent directly to her local pharmacy so she can begin treatment immediately. After obtaining the results, I sent it to Mrs. Anderson's MyChart and made a call to explain the results to her. The gram staining test identifies between gram-positive and gram-negative bacteria based on the structure of the bacterial cell wall. After performing the test, I detected under the microscope the bacteria appeared as a pink stain, meaning it is a gram-negative bacterium. This is significant and helpful because Borrelia burgdorferi is a gram-negative bacterium. After a few days incubating the sample on the macaque agar plate, the plate appeared to be brown, indicating that the bacteria does not ferment lactose. This is a trait from Borrelia burgdorferi, since it does not ferment lactose. This bacteria undergoes glycolysis for ATP production, utilizing glucose from the host body as opposed to fermenting lactose. Considering they do not include fatty acids, amino acids, or nucleotides, it depends on the transport of nutrients from the host. In the Kirby-Bauer test, I assessed the bacteria's sensitivity to several antibiotics. After reading the values of the Kirby-Bauer interpretive chart, results show that doxycycline, amoxicillin, and ciprofloxacin acetyl were the 3 most sensitive and effective treatments. Doxycycline is commonly prescribed as it targets bacterial protein synthesis. This prevents amino acyl tRNA from binding to the ribosome, stopping the production of proteins for bacterial growth. Amoxicillin and ciprofloxacin acetyl function to inhibit cell wall synthesis, weakening the bacterial structure overall and leading to its breakdown. A factor between these three antibiotics is that they are known to treat Borrelia burgdorferi. Considering Mrs. Anderson is not pregnant and does not have any contraindications to these medications, I explained to her that I would prescribe to her one of these antibiotics as her treatment. According to all the lab results, Mrs. Anderson has Borrelia burgdorferi. Although I have distinguished the bacteria as gram-negative, it differs significantly from other gram-negative bacteria. Borrelia burgdorferi lacks lipopolysaccharide A, LPS, on its outer membrane and instead has immunoreactive glycolipids. However, this can be difficult to distinguish, considering it does not have the standard outer membrane of lipids, making it harder to identify under counterstain with safranin. Not only did I look under the microscope to identify the bacteriogram staining type, but I observed the shape. I noticed that Borrelia burgdorferi is a spirochete, meaning it is a corkscrew-shaped bacterium. Additionally, it is pleomorphic, indicating that it changes shape and sizes in response to different conditions. And this bacteria can move using flagella, the structure between the outer and inner membranes to help them move. And they specifically move in a twisting motion, which is a spiral shape. And on top of that, under the microscope, it is typically seen as a single cell. Borrelia burgdorferi is anaerobic, yet it has metabolic limitations. They rely only on glycolysis, the breakdown of glucose for ATP production, for survival. Since they do not encode fatty acids, amino acids, or nucleotides, they are dependent on the transport of nutrients from their host. Since Borrelia burgdorferi engages closely with host tissues, it leads to inflammatory responses and symptoms of Lyme disease. Additionally, a tick is not part of the human microbiome. Their normal niche is staying in specific environmental conditions such as wooded, grassy, and bushy areas, including animals. They usually live in rodents, deer, dog, or cats. And humans are accidental hosts for this bacteria as they infect humans when transmitted through tick bites. Therefore, it is not a human microflora. Lyme disease is a bacterial infection caused by the bite of an infected black-leg tick. Considering Mrs. Anderson was in New Hampshire, she was most likely to be bitten by a black-leg tick as they are located in that area. Ticks are a type of ectoparasite, meaning they live on the surface of other organisms, in this case, Mrs. Anderson's body. When a tick bites, it releases saliva, which is an anesthetic-like substance that numbs the skin when an individual does not feel the bite of a tick. Ticks go through 4 life stages: egg, larva, nymph, and adult size, but they only acquire from female ticks. Given that nymph ticks are smaller than adult ticks, there's a higher risk of getting Lyme disease as it is difficult to see them compared to adult ticks. Although Lyme disease is rarely fatal, it can become a serious issue if it is not treated quickly, and it can cause long-term effects on the joint, the heart, and the nervous system. Despite this disease being spread through the bite of an infected black-leg tick, it is not transmitted through air, food, water, or sexual contact. In terms of being bitten, any age group and population are susceptible. However, people who spend plenty of time outdoors, such as hiking, golfing, or other outdoor recreation, especially in wooded, grassy, or bushy areas. That being said, children ages 3 to 14 and adults over 50 years old are at higher risk. Although Lyme disease can occur in anyone, there is a higher chance for those who are often outside. Based on Mrs. Anderson's symptoms and rash, she was diagnosed with Lyme disease. After carefully assessing each antibiotic and its benefits, she will start on an antibiotic pill called doxycycline, which is highly effective in treating Lyme disease, especially in its early stages. She will take this antibiotic for 14 days, twice daily. It should be the first thing in the morning and in the evening before going to bed. I chose this one because I do not want her symptoms and rash to worsen. To prevent the later stages of Lyme disease, prescribing doxycycline would be the better choice as it inhibits bacterial protein synthesis. This inhibits the growth of Borrelia burgdorferi, hindering the symptoms and signs such as joint pain, fatigue, and inflammatory responses. After reaching out to her once again, I informed her that while taking doxycycline, it should be taken with food, but she should avoid dairy products because dairy reduces the absorption of the antibiotic, avoid refined sugar as it is immunosuppressive, and prevent consuming gluten as it is inflammatory. I suggested that her diet should revolve around vegetables and chicken, for example. Also, I told her that doxycycline may cause her skin to be more sensitive to sunlight, so she should stay away from direct sunlight for the time being and use sunblock with at least 15 SPF. And some preventative advice I give to Mrs. Anderson for the future is to avoid tall, grassy, and wooded areas. And if she ever goes hiking again, she should stay in the middle of trails, wear long pants, long sleeve shirts, to be specific, light color clothing, since it's easier to spot ticks, and wear closed shoes to prevent the tick from attaching to the skin. It was a pleasure to treat Mrs. Anderson. I told her that if she has any concerns or notices of new symptoms, she could call my phone number that is provided on my chart. I told her to not hesitate to reach out, and I provided her links on information about Lyme disease, including its symptoms and causes, and the antibiotic I prescribed to her through Mayo Clinic on my chart. Additionally, I provided a PDF of a pamphlet on Lyme disease for her to read on my chart.